Hello, everyone. Welcome to the Croccast podcast. I'm your host, Nate, along with my co host, Matt.
Hey,
And today we're joined by Zach Loughman of West Liberty University and one of the co hosts of the Colubrid and Colubroid podcast. Zach, welcome to the show.
Hey, y'all. Thank you for having me. Happy to be here.
awesome.
Yeah, so, Zach, you mind telling us first how you first got into reptiles and how you basically got to where you are now?
That takes about an hour and a half. Um, so I'll give the abridged version. Um, there's there's never been a time where I wasn't into animals. I'm a college professor, so I work with a lot of young people, and uh, it's been kind of eye opening in that I get a lot of students who kind of realize they want to work with animals or do ecology or do research, be a scientist like late in life. And for me, there's never been a second I didn't want to do what I'm doing right now. So uh, basically, I have the kind of standard story. I was a total dinosaur nerd growing up, um, and that lasted until I was in third grade, I believe. And it was in third grade that I my family moved out into the country, and I realized that I really liked hunting down the animals and catching the animals. And you can't do that with a non-avian dinosaur. I guess technically you could catch a bird and you're technically herping, but we're not going to go down that path. Um, so anyway, and yeah, science has always been part of the equation too. So um, I did science fairs when I was in middle school. Through high school, I did what I referred to as nerd camp, which was basically this 3 week long camp. Not really camp, but it was a program where high schoolers that knew they wanted to be scientists could live at the college that I teach at now, West Liberty, for 3 weeks. And so I did a salamander project then, and that's when I realized, like, okay, yeah, this is definitely what I want to do, because in that project it wasn't just chasing down the animals or keeping the animals in human care, but it was rather like studying a specific aspect of their biology. And I ended up in that experience realizing that that was definitely what I liked doing the most. um, and so I had an advisor here at West Liberty named Bob Gordon, who was a snake guy. Every city has the snake guy, and he was Wheeling, West Virginia's snake guy. So he kind of took me under his wing, and I am forever grateful for that. Um, and so I went to university here at West Liberty. Um, had a wonderful experience as an undergrad here. Since Mr. Gordon was more than an advisor, he was essentially my grandfather, and he opened up the zoology lab and said, "Fill it." And I was like, "Excuse me?" He's like, "Fill it with animals." I was like, "Oh wow, cool." So all through college, that's when I got into herpetoculture big time, and kind of did the eclectic thing. That was in the late '90s, where there were definitely people breeding, but it was more about keeping. So you had a collection that was essentially one of this, one of that, one of this, one of that. So I had all kinds of monitor lizards in the lab, lots of different species of turtles, snakes, all over the flipping place. And while I was here at West Liberty, I got the taste for herpetoculture, really liked it, but knew that I was much more field based. So I knew graduate school was in my future, and I knew I was going to study snakes. And met my wife here at West Lib. We got married after like a month after we graduated, and then we initially went to grad school at Florida Tech in Melbourne, Florida. And I, of course, went down there for the snakes. Uh, the school was not a good fit for Kathy and I, but it was the best herping of my life. And it just really kind of sealed the deal, because by that point I had done the keeping piece, and I had done the field piece, and the field work was definitely the more fulfilling of the two for me, from as far as a profession was concerned. And so we bounced from Florida Tech in Melbourne to Marshall University, which is in Huntington, West Virginia. And I'm a proud alum of the Poly Lab, which was the herpetology lab there. And my 2 years getting a master's degree were without question, they were among the best 2 years of my life. I could not accept that people could go to school and get paid to go herping. Like that was my job, was to go herping. Like my mind was blown that this was my gig. And I learned how to be a scientist there, and learned a lot about how to do surveys, how to set up a survey, how to work with endangered species, how to plan a week-long collecting trip. All these kind of things that you don't think about when you can only herp on the weekends. When it's your job to go herping for days on end, it adds an order of magnitude that a lot of people don't get to experience. And I was just doing this day after day after day after day. So while I was at Marshall, though, that was in the early 2000s. And that was when Steve Irwin and Jeff Corwin and all those guys were exploding. I was at Marshall from 2003 through 2005. And I saw the job market, and I knew the job market was actually saturated with herpetologists. And my advisor, Dr. Polly, we were driving home from Southern West Virginia, and it is impossible to get anywhere fast in West Virginia. So, you look at the map and it looks like Huntington to this area we were in called Logan was like going to be a 45-minute drive. It ended up being like 2 hours. And so we're just driving on these roads, and Polly said to me, I've been watching you. I know you think it's herps, and I'm not saying don't be a herpetologist, but at the same time, pick a group that nobody studies and be the guy. Because if you can do that, you're going to get funding. And if you get funding, you're going to get a job. I was like, I will listen to you because you have wisdom that I do not have. So we were doing a study along the Ohio River floodplain looking for smallmouth salamanders and streamside salamanders, Ambystoma texanum and Ambystoma barbari. And we kept getting all these crawdads in our traps. And I had a friend who had set up a drift fence array around a pond, and she was actually studying Plasma montanus, the mud salamander. And this population around the pond was kind of unique in that they appeared to do some kind of migration, which is weird for them. They're normally, they're certainly not an ambystomata, but the fact that they were moving in and out of a pond was kind of interesting. Well, one spring night, it rained a lot, and the buckets filled up with not just salamanders, but a bunch of crawdads. And Vanessa was her name. She brought the buckets in, was really pissed, dropped a whole bunch of words that rhyme with the word truck, and then said, figure out what these things are. I was like, okay, I will. And I opened up the bucket, and there were like 50 of these big, gnarly crawdads staring back at me. And that is essentially the combination of the Texanum barbari study and the drift fence array. That's when I kind of got the taste for crayfish. And so I kind of diverged away from herpetology for my PhD because I knew that if I went this route, it would be very lucrative for me. So I kind of got away from herps for a little bit, taught herpetology here at West Liberty multiple times. But in 2016, and a lot of people that have heard my story on podcasts know, my university decided they were going to do a zoo science major. And we hired a guy, and that guy is now the director of the Oglebay Good Zoo, but he was not the director of the Oglebay Good Zoo. That's the zoo that's down the road from us. And so we set the major up, and the idea was that I was going to be the ecology guy, and Great House was going to be the zoo guy, and then he became the zoo director, and my chair came to me and was like, you're the zoo guy now. And my response to that was, excuse me? I'm the field guy. But I thought, what the hell? I mean, people are literally asking me to build a reptile collection and do it the right way. And so I had gotten back into herpetoculture a little bit in 2014. But in 2016, it was just basically throttled down. And my worlds have now converged because now I'm back doing herpetoculture. I do have some herpetology projects, but it's kind of like a gumbo of herps, if you will, my life right now. Because I teach an undergraduate class in herpetology and herpetoculture. I teach a graduate level class in herpetology and herpetoculture. I have a lab that has graduate students called the Evidence-Based Herpetoculture Lab. I have graduate students that are studying the ecology of queen snakes, because the two universes come together with a queen snake. You got a snake that only eats crayfish. There's like no way in hell I'm not studying it. So that's kind of where I am now. but it's been a strange herpetological renaissance in my life, which began in 2016. So that was a little bit, windy, but that's kind of the way the story goes, I guess.
No, that's good. I actually have two questions off of that. So, so one thing that's nice about us is like I'm more into like the research side, and then Nate is more into the herpetoculture side, and we like both, but that's more of our expertise. So it's cool to see that you've kind of had your hands in both. That being said, for someone that wants to go into like more of the research side, how would you recommend them going through that? And then two, along with that, what are your thoughts on like between getting like a master's and a PhD? Like, should you just stop at a master's, or should you get a PhD and that kind of thing if you're wanting to go into like herpetology or research in general?
So there's two questions there. I'll answer the first part now, which is how do you go about getting into herpetology? I actually get this a lot because I tell everybody that my goals are to be an okay scientist, a badass naturalist, and the best teacher I can possibly be. Like that's my trifecta.
Yeah.
And so obviously with herpetology to make it a career involves a lot of education. And so I tell everyone, and this isn't just because I'm at a little liberal arts school in Northern West Virginia that only has 2,500 students, but for your undergraduate, I think it's extremely important that you find a program where you are not just a number, that you are going to get some personal attention by the professors, and that you have professors that are engaged. At a lot of schools, not all schools, I'm saying a lot of schools, undergrads are the paycheck that drives the institution. So they want to be able to say they've got 20, 30, 40,000 undergrads. But if you're a professor in that environment, a lot of times, and I can speak for this perspective because I'm a professor, you live in this publish or perish environment, and you basically have to bring in grants to get tenure. You have to publish papers, and you can certainly do that with undergrads, but it's much more lucrative for you to put your attention into the graduate students, because the graduate students are literally writing a thesis and a dissertation. And so when you get that funding in hand, you can then turn around right into a PhD or master student and make their thesis be some of your future publications. And I have a graduate lab in crayfish biology and herpetoculture, and I can tell you, every single kid that publishes anything in my laboratory, I am going to be the last author on all those pups. So that's kind of the way that I can ultimately have a lot of scientific output. If you're trying to learn how to be a scientist in that environment, you're probably going to be working with postdocs. You're going to be working with graduate students a lot, but getting the professor's undivided attention, that's going to be difficult. So what I recommend people do is that you focus your undergrad on a smaller school where you will get that attention, because I can flat out tell you, being the guy who takes on graduate students, I don't care where you get a degree. What I care most about is the letter of rec. I care about what the professor says. I care about that boots on the ground experience that you have with your professor. And every student that I've taken into the crayfish lab specifically, I have contacted the letter of rec, the person who wrote the letters of rec, and talked to them as an individual. Like I go one step further, because it's an investment if you're taking on a graduate student of time, energy, and funding. So you want to make sure you're getting the right one. I feel like you really can get that attention at the mid to smaller schools a lot more. And I've noticed with my grad students, the ones that come from that kind of smaller environment that did have a lot of undergrad research experience, are the ones that hit the ground and just shoom, go off to the stratosphere. The ones that are coming from the giant R1s that didn't necessarily do anything beyond a capstone project, I have to spend a lot of time getting them up to speed because they didn't have that individual attention. The way to combat that is, if you're at the big school, make sure you do internships that are with, in the field with field people, because you can totally get all the experience you want as a researcher over an internship with the Fish and Wildlife Service or State Wildlife Association or Nature Conservancy or any of those, and make up for that lack of attention you might not be getting if you're at a bigger university. Okay. If you're at the bigger university and you're getting the one-on-one attention, that's fantastic. But the most important thing that you do is you get into an undergrad position where you can do field research, because I can flat out tell you, if your view of what it is to be a field herpetologist is based off National Geographic, that is just not true. That's just simply not true. Great example. I'm writing a book, The Complete Guide to Hognose Snakes, and I am an field guy here. I will not send that book off to get published until I have held in my hand every heterodon species, and I have to go out into the field to find them. I just invested $2,000 in a trip to Kansas, and I was in Kansas for 10 days, and we logged in over 100 hours road cruising, found 35 species of snakes.
Awesome.
But we averaged about a snake an hour if you average it all out,
right?
And not a single Western hognose snake. So, I think that it's important that people understand that being a field biologist, period, I don't care if you're an ornithologist, mammologist, herpetologist, whatever, it just involves you having a lot of grit and being able to deal with the fact that you are going to go out and look for these things, especially rare animals, and there's going to be a whole lot of zeros. And being able to deal with that is a very important element of being a good biologist, in my opinion. But no, that's that. And then the other thing I would say is just read everything you can. Today is a completely different world than when I was in undergrad and graduate school. We did not have ResearchGate. We did not have Sci-Hub. We did not have the ability to find a journal article through a Google search and then get it. So there's really no reason why anybody, if you just learn some of these little tricks, can't start reading books, articles, monographs. I tell all my students here at West Liberty, when it came to becoming an astacologist, a crayfish biologist, there's no class in astacology. I had to learn all that stuff. And the way that I learned how to describe a species or learned how to do a mark recapture study, or it was all through trial and error and just reading. I read everything. And I accepted the fact that the first, second, third, fourth time I went out, tried to do something, there's a good chance that it was going to fall flat on my face. I mean, it's just going to be a total freaking disaster. But if I walk away with one or two little nuggets of knowledge I didn't have before, then it's still a success. And that's a huge part, I think, of being a herpetologist, a field biologist, is that. What was the second part of the question?
The second part was, what are your thoughts on, because I know there's debate between masters or going on and getting a PhD.
Yes. This is a weighted question, and I am, in a lot of ways, people call me the anti-PhD. So I got hired at West Liberty with my master's degree. And I was supposed to just be an instructor. And then my dean walked down the hallway one day and was like, "Hey, you want to keep your job? Get in a PhD program." I was like, "Yeah, great timing, because my wife's pregnant." So anyway, I had to do a PhD in a completely atypical way. It was remote. This is way before the world we live in today. But as far as the PhD versus the master's, I tell everybody you have to get a master's. If you want to do herpetology outside of a zoo setting, just accept the fact you're getting a master's. Because if you want to be a state wildlife biologist, you're going to have a master's. A lot of the state wildlife biologist positions now are being filled with PhDs. So if you don't have that master's degree, you're going to be in trouble. As far as getting a job is concerned. If you want to be a consultant, which a lot of people don't know that that's a job, but environmental consultants are the people that when you're putting in a bridge or you're putting in a pipeline and it goes through endangered species habitat, you can't just put the pipeline in. You got to make sure it's not going to wipe out the crawfish frogs, or it's not going to wipe out the eastern diamondback rattlesnakes or whatever. So you send in consultants who do a complete survey of the area and basically determine whether or not there's the endangered species are there, and then they have to mitigate that threat. You can get those jobs with a bachelor's degree, but I know quite a few people that work for consulting and are in charge of consulting firms, and they all say masters, 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 because with a master's degree, you write a thesis. And when you write a thesis, you learn how to write. So for those jobs, you're going to be writing these great big reports. So, there's that. If you want to get into the Fish and Wildlife Service, you need a master's degree. You can get in with a bachelor's degree, but if you have the MS, you're good to go. And there's actually programs with Fish and Wildlife. One of my students did this. They have an apprenticeship where right at the end of your master's, you don't graduate yet. You go and spend 3 or 4 months with the service. They pay you and they teach you how to be one of them. And then you graduate and you basically go to the top of the list to get a job. And those are nice jobs. Those are high paying jobs. And you're going to be working in conservation, which is kind of the goal for all of us that do this. For the PhD, it's a wonderful, you learn what you are capable of doing when you go and get a PhD. That's what I got out of my PhD more than anything. A PhD makes it so you can teach. If you have any aspirations of being a professor, just get the PhD. Just suck it up, accept it, get it, move on. But a PhD is also going to get you a higher paying position anywhere you're going to be with a PhD. So, there's that. That being said, while you're a PhD student, most PhD students are kind of somewhat destitute. So, you have to be willing to eat, have no life for 3 or 4 years. And it really is an intensive situation. You are literally creating scientific fact that did not exist before you did your dissertation. And with a master's degree, you do that, but you might come up with like one publication. Most PhDs, you're not defending till you have at least 3 to 5 pubs. And some people love writing. I happen to love writing. Other people hate and loathe it. If you don't like writing, PhD might not be the best avenue for you. So you kind of have to do some soul searching. And the other thing I would say, though, to anybody that's considering any of this is just make sure that whatever grad program you go into, the project you're working on, you have to like it. If you don't like that project, the amount of time and energy that you put into that project, it's just going to be misery. If you love what you're doing and can nerd out and geek out and never really stop thinking about it, be the best time of your life. So that's why I say, like, my master's degree was awesome. So yeah, that's kind of a long-winded answer there, but that's my 2 cents on it.
No, that was great. I like to talk to people about that, especially because I'm working on going to get my master's soon and stuff. So, and everyone has a different opinion on it. So I like to hear, what everyone thinks, especially from someone who actually is a teacher and stuff like that.
The one thing I will say about being a grad student is we talk a lot about work-life balance in 2021. And a good graduate student, I'm not saying you don't have a work-life balance, but you are going to be thinking about this stuff a lot. Doesn't mean that you have to think about it like 24 hours a day, but grad school is not especially in ecology and conservation. It is not the kind of gig where you go in at 8 a.m. and then you're done at 4 and you punch out and then you go do something else. Like if you're studying an animal and that animal has a certain activity period and that's it, you have no life during that activity period. That's when you got to get them. Not to bring it back to crawdads, but the field-based research I have is with crayfish. And I have 2 grad students right now who are studying a crayfish. We got big grant in South Carolina to go down and study this group of crayfish that are called Distocambarus. That's the genus name. And these animals live in sandhill upland environments that during late spring through the fall, when there's no rain, it's all sand and a little bit of clay, and the sun bakes it like concrete. And these things live in burrows. But from like Valentine's Day to Easter, when it's rainy, you can catch them. And my 2 grad students are literally relocating to South Carolina from Valentine's Day to Easter. And I have explained to them multiple times, when the rains come, you're out. So if they don't come until 10 o'clock at night, you are out at 10 o'clock at night until 4 in the morning, because you don't do that, you're going to end up writing a thesis that says these animals are very rare. And in reality, they may not be rare at all. Their perceived rarity just might be an artifact of improper sampling. So that's an aspect of this. But, if you're a herper and this is what you're doing, this shouldn't be misery. This should be almost like you're living the dream. That's how it was when I was at Marshall. Like, Dr. Pauly would walk down the hallway and be like, "It's raining, go." I'm like, "What do you mean go? Where am I going?" And he would be like, "Go to the High River floodplain, go to this county, go to this county. I want you driving all night. Let's look for the salamanders on the road." And I pinched myself. I'm like, this is really happening. And I would miss class, and it was okay, because as a grad student, that's your main objective in life is going to be research. So now it was kind of funny because it kept raining on Wednesday nights. That's when I had con bio, and it was a night class. And I think it was the 5th week in a row where it was raining. And he came down the hallway. This is one of my favorite moments in grad school. And he's like, "Where have you been?" Like, what are you talking about? You've missed like 5 weeks of class. It's like, you keep sending me out to look for these damn salamanders. It's like, "Oh, yeah, that's where you've been. Oh, well, it's raining tonight too, so you need to go again." I was like, okay. So, that's that. Now that I have a million grad students, I totally understand why he forgot that he had sent me out, because every day is like 500 individual conversations. But that's the reality of grad school.
So working with, I don't know a whole ton about crayfish, but being like a herp guy and have working with salamanders, and then now that your expertise is in crayfish, have you ever come across working with or come across just chytrid fungus disease with that?
No, not so much. The chytrid fungus issue with herps and crawdads is primarily associated with the red swamp crayfish, and it's also mostly associated with populations of herps where red swamps have been introduced, and basically the chytrid that's found here in their native range in the southeast is a different strain or genetic line is introduced to a place like California, and then you see issues. What I actually run into more than anything is a whole bunch of really interesting reptile and crayfish associations. So, example, I've dug a lot of crayfish burrows, and when you get down into the southeastern U.S., digging crayfish burrows is really kind of it's fun, because you can bump into snakes and frogs and all kinds of things down in the burrows. It's great when I'm digging with like other crayfish people that aren't necessarily herp people, because when they find the snakes, they like freak out, and I'm like, Christmas morning. So
right,
yeah,
right.
But anyway, but like, I know that the crawdads are the burrows that they're making. I think that crayfish are critically important for maintaining a lot of herpetofaunal communities in the southeastern United States, because they create these areas where the animals can get away from heat. And I think more importantly, since they burrow to the groundwater table, and I don't think herpetologists have really kind of dove into this, and it's not their fault, they're not studying crayfish, but I think that there's an awful lot of water resource potential where we perceive it to be a drought, and these crayfish are digging down to the water table, and things like ground dwelling lizards and snakes and animals like that are actually able to go dive down the burrow and actually get access to water. Because when it's dry outside and we're on the surface and we start digging burrows, you start pulling frogs out of those things hand over fist, because it's a water refugia for them in a drought. So no, that's totally an area of future research for me. I just have noted everything. I wrote up an observation with the Gulf. I don't remember the common name. It's Regina rigida or Leiodites rigida, depending on your taxonomy. But it's one of the crayfish snakes, and I was down in near Mobile, Alabama, looking for a crayfish, or very rare crayfish. And we were in the colony, and just right on the surface was a dead crawfish snake. It's like, what the hell is this? And then I picked it up, felt a lump, and of course I was going to, dissect that out and see what it had eaten. And it had actually eaten this endangered crayfish, which was a burrower. So in that instance, if you didn't know that that was a crayfish that lives in a burrow and it's not one that just lives out in the swamp, you wouldn't realize how big a deal that was. But that crayfish, that snake, almost certainly had to go down in the burrow to pluck the crawdad out instead of go out into the swamp and get the things we normally associate them with.
So if that's true, that they're burrowing down to the water table and all that other stuff, could you make an argument then that they could be more important than a gopher tortoise? Not to hate on the gopher tortoise, but
I think they're just as important.
this is
Yeah, without question. Gopher tortoises and crawdads are doing the exact same ecological function. They're creating a complex, heterogeneous habitat that in their absence is not present. And it's an underground habitat. And as soon as you get a meter underground, you get kind of the constant climatic regime, which basically is going to be utilized by anything that's ectothermic or cold-blooded. So I just think that the gopher tortoises obviously are digging a bigger burrow, so they're going to be impacting bigger vertebrates.
right.
And the crawdads are digging a smaller burrow, so they're going to be impacting smaller vertebrates.
More of an opportunity for snakes.
Mhm.
And,
yeah.
Is there? And this may not be something that you guys have looked into yet, but is there a certain size that they have to reach before burrow is being able to be utilized by a snake?
I think at least a centimeter diameter to the burrow opening. We found that same trip that we found the rigida, that at the adult, we dug up quite a few young of the year rigida right out of crayfish burrows that were about a little bit bigger than the diameter of a pencil eraser.
Wow.
We also dug up amphuma, which was really cool.
Yeah.
Amphuma means were kind of up in the crayfish burrows too, and they were itty bitties, like fresh out of the egg amphuma, which was really, really neat to see. So, yeah.
So, a bit less crayfish oriented, but you mentioned that you have for your zoo program, you kind of built your reptile collection at the zoo up from the ground level, and you especially emphasized that you built it correctly. So, you want to go a little bit into about that collection, your work with that?
Yeah. So we knew at the onset of the zoo science degree, I knew that I wanted to have that it was extremely important if we were going to be training zookeepers that they get as much experiential learning as possible, which is fancy education speak for I don't want to just kill them with PowerPoints. If we're talking about how to hold a snake properly, I want to have multiple kinds of snakes to hold. Big ones, little ones, medium ones, calm ones, psychotic snakes. Like basically, if you only learn how to pick up a ball python and then you're tasked with picking up an angry crebo, that's like a whole world of difference. So I knew we needed to have a diverse collection of animals. I also knew it was going to be reptiles for two reasons. One, that's my background. And if I'm going to be in charge of something, I got to like it. So we're going to stick with the animals that I feel comfortable with. And two, if you start building up your numbers of mammals and birds, you have to get USDA permits. And I just didn't want to deal with the USDA permits. I don't mind dealing with the USDA permits for a handful of mammals, but when you get up, around 10, 20 different mammal species, you got a problem in permitting. And you need time. And I don't have time to do hardly anything, it seems. So I knew that it was going to be reptiles. I knew that I wanted to do a lot of research into the whole argument that's present in herpetoculture, which is the whole rack versus naturalistic. I, as a scientist, look at that argument and I literally see pluses and minuses to both. So I thought it would be cool to get animals that would be active, that would be moving around a bunch, and have animals that weren't moving around a bunch, so we could kind of do some research with that. If we're going to do that, we need animals in replicate, because if you just study one ball python, you can't make a determination for all ball pythons. Sorry. So we were going to have research colonies of certain taxa. And then I also knew I didn't want to have what I refer to as a Petco collection. I didn't want to have cresteds, beardies, corn snakes, Cali kings, and that's it. I wanted to have animals that were a little, like, not necessarily one of a kind, super rare, but things that were, if a person with a herpetological background walks into our building, they kind of walk away like, okay, they know what they're doing. So knowing all of that, we started small and built some built-in enclosures, and we got some of the larger lizard species. So I got a black and white tegu, raised it from like a hatchling up, so it was socialized and it definitely knew who I was, which was good. We purchased a water monitor. So, and I got the subspecies macromaculatus, which is one that gets big but doesn't get huge. But that required a large built-in enclosure, so we had to do that. We did. And then I focused a lot early on geckos. So I knew that Rhacodactylus or Diplodactylid geckos were pretty easy to take care of. But they were also they had some complicated behaviors. So our one Petco herp, if you will, is we do have a large colony of cresteds, because I bred all the cresteds at my house and then kept track of their lineages, and we used them as a mock for a stud book. So basically, keeping track of genes and lineages and all that kind of stuff for the living collection. We have those. We have some gargoyle geckos, and then I purchased a bunch of Leachianus. So we have the great big giant geckos, and we have the island strains or races, and we have the mainlands. And I wanted those because they have very complex behaviors. They set up monogamous bonds. They're a little bit intimidating. They vocalize, and I thought that they would be a good intermediate sized animal to introduce the students to all of that. And then basically a smattering of snakes. I wanted to get as many different types of snakes as possible. So we have pythons and boas and colubrids and colubroids, and it's just taken off. And then over time, we brought on the grad students. The grad students projects, their theses for zoo science are based out of the collection. So I started amassing collections of certain species where the goal is to get to between 15 and 30 individuals. So we have a statistically, a population size that goes beyond normality. So that's been fun. Right now we have a corn snake colony, a hognose snake colony, false water cobra colony, which is a very weird snake to have a colony of, but they just happen to be my favorite snake on planet Earth. So that's why they're a colony. And so, yeah, but everything is in PVC enclosures. We also have large racks, and we do a lot of science with the animals. So, they're not pets by any stretch of the imagination. Every animal in this particular collection has a purpose, and several of them actually have multiple purposes. So that's kind of where we stand right now.
So you mentioned you have that colony of false water cobras. You mentioned that they're your favorite snake in the whole world.
Yeah.
Mind going a little bit in depth about, I guess, what you find so fascinating and enjoyable about them?
Oh dear. Okay. All right. Well, I like them so much that I'm writing a book on them. In fact, it is right here. I was editing it before you guys came on, so they inspired me a little bit. The thing I love about false water cobras is that they kind of check every box that I like in reptiles. So my favorite herps are herps that live in water. I don't know why. I've just always had a strong affinity to creeks, rivers, and swamps. Like, those are my favorite habitats on planet Earth. And of course, falsies go to water. I like animals that defend themselves. I guess that's the professional way of saying, if it tries to rip my face off, I'm giggling and cackling like an idiot as that's happening. Like, that's always been something that I've liked. And initially, I had this perception that false water cobras were that. I can tell you that they aren't now that I keep them, but they will definitely let you know, you're pushing me too much. I'm not comfortable with what's going on. I really appreciate that part of their biology. They're the only snake I've ever worked with that whips its tail like a iguana or a monitor lizard. And when they whip you, like a big 7-foot false water cobra does that, it hurts. It gets your attention.
For sure.
The other thing is that they get to be freaking 7 feet long and weigh about 10 pounds. So this is not a small snake. When a large falsie has the same dimensions as a woma or a black-headed python. Like, they are a big, burly animal. And then the final part is that they have a ridiculous feeding response that you cannot appreciate it until you've observed it. They do have the rear fang thing going for them, which I'll fully admit I do kind of like that aspect of them. And they have an intelligence, and I will use the word intelligence because we've actually done experiments now, and the results of those experiments are indicative of intelligence. So, this is a snake that you can actually train. So we've target trained our snakes. I have a graduate student, this was her thesis. She can shift the false water cobras from their bins into a feeding bin by presenting a target. You can do that with other snakes. It takes them a while. The problem to learn it, individual false water cobras can learn this in an incredibly short period of time. But falsies are so independent as animals that they will just choose not to learn it. Like, they know what's going on, they just don't care. I really like that defiant aspect of their biology. So, yeah, they're easy to breed. They're fun to feed. They kind of just check all the boxes for me. I did not, back prior to starting Zeusai, if somebody asked me what my favorite snake was, I would tell them, well, I really like Nerodia, and I really like this, and I have this favorite, and it's my favorite pit viper, and this is my favorite python, and this is my favorite colubrid. I didn't really have a favorite. Now I do have a favorite instead. And I'm going on record with your podcast. If I do not make it to Argentina or Paraguay to find one in the wild before I die, I will die unhappy. I have to herp one of these things. Like, I can accept spending all that time in Kansas not getting a Western hognose snake. I will get my Western hognose snake. I have to find one of these things in the wild. There's just no way. I don't want to keep it. I want to watch it, possibly capture. I don't even know if I have to catch it. I just want to see it doing its thing out in nature. Like, that just has to happen.
So. I'm the same exact way when it comes to stuff like that. I gotta see, like, if something cool, I gotta go see it in the wild. Like,
yes.
That's just the way I just gotta see it. It's so cool.
Yeah.
So going off of that with rear fangs and whatnot, I was listening to these one guys. They were talking about rear fang snakes, and they were saying that they didn't think people should handle rear fang snakes like hognose and whatnot, for the venom aspect and stuff like that. What are your thoughts on that?
That is a great question. So given that I'm writing the book on, so the first book I'm writing is on South American dipsadids, which are it's a group of, it's actually a family of snakes that people don't really recognize as a family, though all the scientific evidence of the world supports it. And hognose snakes are in that family Dipsadidae, which a lot of people don't realize. That's why we named our podcast, by the way, Colubrid and Colubroid Radio, because I'm such a freaking nerd that I couldn't have my podcast called Colubrid Radio and be talking about a false water cobra all the time. Like, no, we're going to do this right. So with these dipsadids, I can speak to the dipsadids. The dipsadids have an awful lot of these things called metalloproteases in their Duvernoy's gland secretions. And metalloproteases are a group of enzymes that are present in viperid venom. So the reason why when a timber rattlesnake bites you and you go, ouch, that hurts real bad, and my hand is turning to goop, one of the main drivers of that enzymatic action are all the metalloproteases. When you look in the Duvernoy secretions of most dipsadid snakes, they have the exact same metalloproteases. So at that level, it's kind of black and white. If they have the same thing a viper has, by all means, we should not be holding them. But then that begs the question, when you see some buddy getting bitten after they boop the snoot of a hognose snake, which that statement, by the way, I'm just going to stop because I'll start using the words that rhyme with truck if we start talking about booping snoots. I hate that. But anyway, if you do that and then the hognose snake goes like nom nom and bites you, some people have nothing happen to them. Other people get these massive lymphatic edemas and, they absolutely have something in there. Well, why? Why does one person have it, one person doesn't? The problem is in the venom delivery. A viper has a closed venom delivery system and it's pressurized. So basically, when a viper bites you, the venom's going to go down the venom gland, down the venom duct, down the fang, and come out one hole. So that's all contained. With these dipsadids, some of them actually do have lumens and things in their Duvernoy's glands, but most of them are just exuding it out onto the surface of these teeth in the back of their mouth where there's a groove. And then it's going to kind of go down that groove, mix with the spit of the snake, and it's not pressurized. So the venom delivery is exponentially less involved than it is with a viper. So what ends up happening is most people will get bit and nothing happens, but most is not all. So then someone's going to get nailed and it's going to be bad. So I am on team use hooks, use gloves. Like, I know that there are people that listen to this and be like, what the hell is he talking about? It's a hognose snake. If you actually do the research and look into Western hognose snake bites, like the journal articles that go over, like there's this genus called Toxicon where they do a lot of bite histories. And I've read the plains hognose snake bites or Western hogs. Some of those things are horrific. Like if people, like we're talking great big blood blisters that pop. And then you get secondary infections and then it gets necrotic. Like the same thing you see with a viper. It's just 1 in 10 million people or something crazy. But I don't want to be that 1 in 10 million. So, there's that. And it's really kind of funny because some rear fang snakes have this perception of being extremely hot. I'm not going to talk about which ones that in reality are not. And others we kind of think of as just, they're not that dangerous. We can, hand over hand. And they actually have a pretty significant Duvernoy secretion in there. So I say hooks all the way. What's it going to hurt?
Yeah.
And the thing that people need to realize, especially keepers, I was on social media today and I had to put the phone down because there was some guy free handling a Bushmaster because that's wise. But like he literally said, like, what's it matter? Why does it matter if he's doing that? Leave him alone. This is why it matters. When that guy gets bit by the Bushmaster or some little 6-year-old boops the snoot of the hognose snake and then gets latched onto, and then her finger becomes extremely necrotic, somebody in a city council, some legislature guy, someone who's making rules is going to see that and be like, wait a minute, they have these things? Well, they shouldn't have these things. And that's what leads to this overregulation. If we're always putting our best foot forward, we keep that from happening. And that's why I think it's so important for people to just respect the snakes. It's that simple.
When I first heard about that with the hognose snakes, I had the same reaction. I was like, "What is this guy talking about? It's just a hognose snake." And then so then I went and looked into the bite stuff, and I was like, "Oh yeah." So since then I've been more team like hooks with that kind of stuff.
Mm-hmm.
Why? So since it's an open system, it's not pressurized. One, why is it like that? Why did they evolve to be like that? And then two, the people that aren't having any symptoms, is it that they're just not getting envenomated, or is it they're just not having a reaction to it?
Yeah. I'll do the second one first and then the first one second. Yeah. The people that are not getting reaction, this just gets into all the individual variation within a population. It's classic biology. All of us on this podcast, anyone listening to this, we all have a different set of genes that control our response to foreign agents that enter our body. And some people are hypersensitive. And so if someone's hypersensitive to any of those molecules that are present in the Duvernoy's secretion of a plains hognose snake, they're going to have a much bigger reaction than somebody who does not necessarily have hypersensitivity. But what can happen with rear fang snakes, which people do not realize, is if you work with them day in, day out, you can absolutely have the same effect people have that work with vipers and elapids day in, day out. You're going to be exposed to those proteins because the spit in their mouths, it wipes off on the substrate, wipes off on the enclosure, and then you can actually build a hypersensitivity because you're always using your antibodies in your immune system to combat the toxin, and inevitably you reach a point where you use it all up. That's a really primitive explanation for what's going on, and there might be someone listening and like, it's not quite like that, and that's true, but essentially that is what's happening. So in that situation, you can get bit and have a really bad reaction. So like I have the water cobras, and we produce a lot of water cobras for experiments, and I have to sex these things, and I have on gloves and things, but even then the neonates they're insane, and they're whipping everywhere, and I've totally been bit on the wrist like 2 or 3 times. Nothing has ever happened to me, but I know, given that they're right there. I sit in this office all day long with them. I'm being exposed to that. I'm going to have a reaction sometime. You know what I'm saying? So that's kind of how that works. With these snakes though, if they bite you and they are able to really get a hold of you and work in the secretions, that's when you're going to have a really nasty. If you're going to have a nasty reaction, that's when you're going to have it. So, and the other question was, why is their system built that way? Basically, yeah. So that's also a very interesting nerdy discussion. The rear fang condition has evolved independently in multiple lineages of the advanced snakes. So if you go by the most up-to-date modern taxonomy, there's a group of colubroid snakes in Africa. They're called the Lamprophiids. They include things like Malagasy hognose snakes, Malagasy cat-eyed snakes, house snakes. Those guys have a rear fang delivery system. There's the family Dipsadidae, which is the most diverse group of snakes in the neotropics in Amazonia throughout South America. Every one of them has a rear fang. You have true colubrids like Boiga in Southeast Asia, the mangrove snake, the green cat-eyed snake, the dog-toothed cat-eyed snakes, or Toxicodrius is their genus name. They have this rear fang condition as well. So in reality, the rear fang venom delivery system is the most common venom delivery system in snakes. It's just we spend so much time focusing on vipers and elapids that we think that they are the main type of venomous snake. In fact, natricines, garter snakes. There's a species of garter snake out west. I can't remember which one it is off the top of my head, where people have been nailed by it. It might be Thamnophis elegans, and have the same kind of reaction you would from being bit by a small neonatal copperhead. So, this has evolved many times. One of the ideas for where it came from is basically to aid in digestion. If you start to digest something while it's alive, you're probably going to kill it. So there's an added benefit of that. And then when you start tweaking out those biomolecules and evolving different strategies for different types of animals, you're going to just kind of serendipitously stumble onto a neurotoxin when you were primarily doing proteolytic things with your venom. And now, boom, neurotoxic venoms show up. But it's much easier evolutionarily to simply take a gland that is associated with your top jaw and historically was dumping saliva and turning that into something that's making a peptide, and then taking the teeth in the back of your mouth and just making them bigger. That's easier to do than to evolve a venom gland, a venom duct, a fang. Like if you think about a pit viper, they don't just have a fang, they got a fang on a hinge. That's there's Selenoglyphus snakes, so that's moving. That's difficult. There's a whole lot of evolution going on there. If you just simply make the teeth in your back of your mouth bigger, that can happen randomly within a population, and it ends up being a beneficial trait, and then it manifests itself ultimately. So, but that's kind of the backing behind that.
So with the burrowing asp snakes of like East Africa and Middle East, be that rear fang, or are they something else entirely?
No, they're their own happy little nightmare. Uh,
Stiletto snakes.
Yes, I saw those in Pennsylvania. A lot of their shows around Pittsburgh have venomous snakes in them, and I was at a show in September, and I saw stiletto snakes on a table. I'm just thinking, why? Why would anybody want that? But anyway, no, they're their own unique fanged snake. They're fantastic, but I don't want to touch one ever. So.
That's interesting. I assumed that rear fanged would be more of a, and this might be too strong of a word, but more of a disadvantage compared to like an elapid or selenoglyphic, snakes and stuff. But to your point, that makes a lot more sense. That it's just much easier to evolve, and it obviously gives you an added benefit to something that's not necessarily venomous.
Yeah, and given the position of where the teeth are, if you can get the prey item back in your mouth far enough to employ those teeth, the teeth also serve this function in a lot of species where they serve as a point of they're going to grab hold of the head of the prey item, and then since they're large and they're oftentimes recurved, so they're going to be facing towards the back of the mouth, it's almost like having fishing hooks in the back of your mouth. Like the frog's not going to be able to wiggle out of your mouth. It's got to get past these large teeth.
So, but you mentioned that you call your show the Colubrid and Colubroid Radio because colubrids have been broken up and reclassified and shifted around a lot more lately. Mm-hmm. So you want to go a little bit more into details about what is a true colubrid, I guess.
Okay, so the Colubrid D, as it was historically known, was, when I teach herpetology, I always refer to it as one of the best examples of a taxonomic dumpster fire ever created. So basically, there's a term in taxonomy and systematics called there's a lot of groups that we refer to as a junk drawer taxa. That's actually like written in formal peer-reviewed papers. And if you think about what your junk drawer is, most houses have one of those, but you open it up and there's staples, paper clips, a screwdriver, baggies, a notebook, tape, like just total random stuff. And the reason why you put it in the junk drawer is you got to have a place to put it, but none of these things really has it's not logical to dedicate one specific shelf or something to that. And Colubridae absolutely was that. And the reason why Colubridae historically was this taxonomic junk drawer is, if you think about it, the colubrids, the term colubrid literally translates to typical snake. So basically, all the snakes that were special in their phenotype got their own families. And what I mean is, like, you look at a boa and a python, there's something different about that snake. The pythons have the heat pits. Boas, many of them get large. They've got the spurs down by their cloaca. So you can look at them and physically see traits that you can use to allocate and put them in their own group. With a viper, you got the fixed fangs, live birth for most of them, egg layers for some of them. Elapids, you got the fixed fangs. It's all well and good. And then you have other things like sea snakes and the acrocordids and the typhlops, worm snakes. So you got all these different snakes that have the very distinct phenotype. But then when you pick up a garter snake and a rat snake, at face value, you can kind of tell they're different. Garter snakes are squishy. Rat snakes are kind of solid. Garter snakes have live young. Rat snakes lay eggs. But at the same time, they kind of have the same general body plan. Neither of them has a pit. Neither of them has spurs. And so what taxonomists did is they would just simply say, you're a colubrid, and put you in the Colubridae. And what a lot of people in herpetoculture don't realize is that Colubridae has been argued about, fussed over since the late 1800s, and all throughout from 1900 to 2000, there were people working on this all the time. And basically what people were arguing was there really wasn't, there were these traits that you could use to separate them out into subfamilies, which is basically their own little groups. And that's where terms like natricines, you might hear people say that. For the longest time, garter snakes, water snakes, queen snakes, Kirtland snakes, line snakes, the Storaria, they were all in the subfamily of Colubridae called the Natricinae, natricines. And the reason why they were put in their own group is it made sense, because while they have a typical body plan, all of these snakes have keeled scales. All of these snakes have a specific type of musculature where they're not constricting prey. That's why they're squishy. All these snakes are ovoviviparous to viviparous, so they all have live young. So we got what we need to put them in their own group. And basically the argument was, is it worth doing? Can we just keep them in as a subfamily, or do we recognize them as a family? And what happened is in the late 90s, early 2000s, people started looking at DNA. And initially they were using the cytochrome oxidase 1 gene. Then they started to add in their nuclear genes. Now they're doing whole genomes. It's crazy. But when you start looking at the DNA very clearly shows that these things are families, that the Colubridae is multiple families, or what was perceived as the Colubridae is now multiple families, and that some of these families, when you look at what historically was what is going to be a colubrid forever, are actually not closely related to them at all, and are more closely related to things like cobras. And so now, today, we have lots of things that were subfamilies that are now families. And just a quick rundown. The family Colubridae, what will forever be a colubrid, it encompasses the large colubrids that are solid in musculature. Most of them are rodentiferous, so they're going to be eating rodents. They constrict their prey, or they eat birds. They're egg layers. Most of them have this weird thing going on where they don't really have well-developed Duvernoy secretions, though some of them absolutely do. And the Colubridae include all the rat snakes, the milk snakes, fox snakes, gopher snakes, bull snakes, the Asiatic rat snakes. They include down in South America Spiloides, the puffing snakes, and Spiloides pilatus. I don't know what the common tiger rat snake. Is that what that one is?
Yeah, my buddy has a pair of those. So
yes. So, but that's it. That's what people don't realize. Like, that's your colubrid. Everything else, if you're going to be a scientist and use your terminology right, is what we call a colubroid. And the suffix broid means to take the form of. So you can totally give credence to the fact that, these things look like members of the family Colubridae, but they are, based off all the best available evidence today, definitely in their own family. So like now, all those natricines, they're in their own family. The Natricidae. The Dipsadids are now in their own family. The Dipsadidae. The one that I think is really interesting, that it just seems like no one got the memo on, but it's crazy, is the family Lamprophiidae, which is the African rear-fanged snakes. And they are actually the ones that are more closely related to cobras than they are to colubrids. They form a clade, or a grouping, where you have elapids and you have these Lamprophiids. And then you got to go back on the tree to a node, and then you drop down, and then you get into the node that has the natricines, the Dipsadids, and the true colubrids in it. So, and a lot of people will say to me, like, why do you care? Why does it matter? I think it matters on a lot of different levels, because if you're talking to scientists, you want to sound like you know what you're talking about. Right. And I can flat out tell you, being a card-carrying scientist, nothing drives me crazier, especially in the past 2 years, than having somebody say, well, I've learned it this way, and that's just the way it's going to be. Well, that's all well and good, but you learned it incorrectly, and let's, adapt and evolve our way of understanding. But it also comes into keeping, because I take this evolutionary perspective with my keeping, and I can flat out tell you, like, understanding what makes a natricid a natricid, and then applying that natural history knowledge to their keeping, makes me a better water snake keeper or a better garter snake keeper. And if I just say, oh, it's a colubrid, then I'm essentially saying it's just like a corn snake, and it's about as different from a corn snake as you can get. Right. So that's why I take I actually honor that stuff, and I think it's important for people to understand it. And if you have 5 snakes at home, you don't have to know all this stuff. But if you're going to be like a serious herpetoculturalist, or you're going to be you definitely want to go into academic herpetology, you got to learn it. Now, what's neat is the verdict is actually still out. There's the younger herpetologists seem to adopt this new wave of thinking. The older guard of herpetology, some do, some don't. And it's been kind of interesting to sit back in the bleachers and watch this, because since I'm, my day job is being a crayfish biologist, but I can take all the bits and pieces I know and now apply it to herpetology. It's been kind of fun. It's actually been kind of nice being in this position and not having to have like an official stance with other herpetologists and things like that.
Go ahead, Nate.
Yeah, but I was just going to say, hearing about that the African rear fangs are more closely related to cobras. I know this isn't 100% accurate, but now it kind of clicks why boom slings and twig snakes pack a lot more punch than a garter snake would.
Yeah.
Interesting. I forgot what I was going to say now. That's one thing that I find funny with herpetology is when it like originally kind of like first came on the scene, if you will, way back when, you have all those guys, which is kind of like the older groups, and then it more recently kind of just exploded. And so you have that there's like kind of a big gap between like the younger people and the older people. And so it's really every issue you can see it kind of divided between the younger people and the older people and stuff. So I like talking to the two groups because it's just interesting to see how they see things completely differently too. So it's really interesting.
Well,
do you have are colubrids your favorite, or clubroids your favorite, or is it just false water cobras that you're really more into with that?
It's definitely colubrids and colubroids. They're my jam. When we got the collection here, I was like, pythons, let's get pythons. And it was really weird. I thought I wanted to be a Morelia person. And I am. I've got poplin carpets, or whatever we're going to call them ultimately, are right here. I have a lot of snakes in my office. And I like them. But for me, it's just, I like, the thing I like about colubrids is I am, in case you couldn't tell, a bit of a spaz. And I got a little bit of ADHD, especially when it comes to biology. And I'm like, my way of doing everything in life is go, 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 go. And having a large, pretty snake that just says like, nah, and sits there all day, that just doesn't do it for me. But having like the Barons racers that are zooming back and forth, or the garters that are very active, I like that. It means you got to do a lot more maintenance, so I'm always cleaning poop. But I'm okay with that. If I've got to clean poop, that's fine, because I'm only keeping things that I'm interested in now. So it's worked out for the better. But yeah, no, definitely my scientific interests are with them. My herpetocultural interests are with them. They're definitely my group of snakes.
Is that why? Just because they're more active than most?
Yes. That's part of it. I like that their taxonomy and systematics is complicated.
Yeah.
In the world of crayfish, I describe new species of crayfish, so I'm like in that up to my hips. And it's nice to take, like, when I do that with a crayfish, it really freaking matters, because I'm the guy writing the paper. So there's this, like, everything's got to be right. I can take a step back and use that part of my brain and read a new species description for a snake, or read about the taxonomic history of colubrids, and it's just fun, because I have skin in the game, but I don't at the same time. So I like that. And then just the snakes that I have always enjoyed the most capturing and going out and finding are colubrids and colubroids. It took me until my mid 30s to realize I don't have any problem going out and catching 30 or 40 northern water snakes or common water snakes in a day. I would rather do that than go off on some long-winded mission and spend 12 hours and find one rare animal, you know? So, no, they just kind of really, I just love them to death. So,
That's fun to listen to because everyone has their own like ultimately what their favorite thing comes down to like what they're looking for most in like a reptile. So like for you it's like movement and stuff like that. For me though I like to handle things. I
yep.
want to be able to handle it. So that's when I look for reptiles that I want to keep and stuff. That's the thing that I look for is like is this something I'd be able to handle and stuff like that. So it's fun to listen to what the different things that
Mm-hmm.
get people excited about it.
Yeah.
Yeah. So what are some of the other things you have in your university's collection other than false water cobras? Like, what are some more unique animals in there?
So we have probably the most unique animals we have are Hispaniolan gollywasps.
Huh.
And these are a type of lizard. They're in their own family now. They were lumped in with alligator lizards and abronia and things like that, but now they're in their own group. And we got those things from St. Louis Zoo. So the St. Louis Zoo had they had a really good year breeding, and they were running out of space. And they basically one of the things I've gotten to do is meet curators in zoos and basically try to form relationships. And so I formed a relationship with the curator at St. Louis, and it's Lauren Augustine, and she essentially said, well, if you're willing to do the mountains of paperwork so we can shift these things from us to you, we will certainly give them to you. So we aren't breeding them. We just kind of have them here, and we're growing them up as part of the project at St. Louis. Other than that, we have some other kind of weird animals. We have a wood turtle that was seized by the New York Department of Conservation. So poachers figured out that the West Virginia population of wood turtles was doing pretty good, and they basically came down in and captured them, and then we're moving them and selling them to China because China's right now really into pet turtles. Turtles are kind of the thing when it gets to herpetoculture over there. And this guy was busted with a bunch of box turtles, and when they were checking his stuff, he fessed up and was like, well, there's some wood turtles over there, and you can't let them go, but there's no sense in like euthanizing them. So the New York gave them back to West Virginia's DNR, and I'm extremely active with our state wildlife agency, and they were like, do you want this wood turtle? I was like, sure, we'll take the wood turtle. And now we're quickly becoming the repository for animals that were seized under those conditions. So we have that. But for the most part, the collection's just kind of normal animals. I also have a collection, my personal collections at my house. And the two collections do merge because we brumate all our snakes at my house and my garage because it's just, my garage is perfect. I have a little area where it gets super cold, and I can drop animals down to the upper 40s, low 50s, which is what we need to do. But I've got a bunch of locality king snakes. So I did have a bunch of Asiatic rat snakes, and it was, you might have heard me say, like, I'm now keeping what I want to keep. I like the Asiatic rat snakes. I still have a couple. I keep Japanese rat snakes and Oreocryptophis, which are the bamboo rat snakes. But I had a bunch of great big beauty snakes and Dion's rat snakes and rhino rat snakes, and I basically realized that I like them, but when I got home, I didn't want something that was involved a bunch of brain power because I was so tired. And so I moved those on to forever homes, and now I just have locality Getula King. So I have not the Easterns, but I have a whole bunch of Florida king snakes from different counties and different phenotypes. I have speckled kings, desert kings, California kings, obviously the false water cobras, Madagascar hognose snakes, tricolor hognose snakes, Barron's racers, Madagascar cat-eyed snakes. I also like short-tailed pythons. I really wanted to be the Morelia guy, and then I got a blood python and was like, yeah, this is definitely more my speed. It's a giant slug that wants to rip my head off. It's perfect. so I now have a pair of bloods. I'm looking to get I have a nice big adult female Borneo, but I'm being choosy with the male, and then I like the black Sumatrans a lot. So I have a trio of those. And then the ultimate water snake. I have a pair of yellow anacondas that are fantastic. Awesome. They're in big enclosures. I had these real expensive, extremely large PVC enclosures built for them that are in my garage. And so, yeah.
What are your thoughts on, going off of that? What are your thoughts on like size of enclosures? So I know I've heard people saying that there's studies saying that like snakes don't need a lot of space and stuff as long as they have, and which is, we've actually had this conversation before with hides. You know, they don't want, like, some snakes want smaller hides and smaller areas. But as far as like enclosure size, what are your thoughts on, like, as far as like size, like just size when keeping?
My thoughts on that is that you have to know the biology of the beast. If you do not understand the natural history and ecology of the snake that you are keeping, or the lizard, or the turtle, you're going to base all of this off of what others have done before you, and that very well might be spot on correct, or we might be consistently keeping things wrong. So a big thing for me, in fact, I have a lab. I'm the only person I know of that has 2 graduate labs, by the way, that don't have a damn thing to do with each other. Because I've got the crayfish lab, and then for zoo science, we have the evidence-based herpetoculture lab. And with the grad students that are in that lab, what I preach is that we're going to go study the biology, number one, and we're going to, if we can't get out into the rainforest, we're going to do a deep dive in the lit. If we don't have lit, because a lot of these things literally don't have anything written about them, we're going to then watch the animals, give them different scenarios. So a barren enclosure, an enclosure with a lot of sticks and like climbing opportunities, an enclosure with hides on the bottom, an enclosure with hides on the top. And then we're going to just videotape them and see, are they using the habitat? And then use all of that to devise our husbandry methodology. That's what I like to promote. I do think for most snakes, it is important for them to be able to stretch out the entire linear length of their body. And it's the reason for that is based off of data that came before the recent present. There's been quite a few papers, they've mostly come out of Europe, that are really pushing this idea where you have to have the enclosure be the length of the body of the snake, or 3 times the length of the body of the snake. And my problem with them is not what they're saying. It's when you read the papers, they have an agenda. Like when you read these manuscripts, they're impassioned, they're not very objective, and they make me as a scientist go, what are you up to? Like you're pushing something here. I want to just see the evidence. And there is evidence that if a snake cannot stretch out all the way, they cannot allow their lung or lungs to fully stretch out and respire properly, that you're going to end up with a problem with anoxia. They're going to get sclerotized tissues on their lungs. Their backbones are going to fuse a little bit. And that's where I feel like they need to stretch out. However, you can mimic that. If you give the snake time outside of its cage to just slither around, you have in effect mitigated that issue. Now, I'm not saying that then justifies keeping a 6-foot snake in a 2x2 box, because that's just wrong.
Right.
But at the same time, like the water cobras that are in my office right now, they're in 6-foot long enclosures. 2 of my big breeder females are 7 feet long. So what do? I open up the damn door and I let them out 2 or 3 times a week. And they're destructive as hell, and they knock everything over in here. But at the same time, they're able to exhibit what we call species-specific behaviors. And so that should be the target, is species-specific behaviors. I have snakes at my house that I keep in a rack. I don't keep them on newspaper in the rack. I keep them with about 2 inches of substratum, a cocoa block mix with some orchid bark and sphagnum moss. And then I have a light that I positioned over the rack so that it's shooting down right in front of the rack. And it basically imitates dappled sunlight because the species that are in that rack are fossorial. They spend all their time on the ground. And that decision to put them in that cage that way was not based off of anything other than me reading all about Oreocryptophis, which are the bamboo rat snakes. Okay. So, like, in that situation, I got no problems keeping that thing in a rack. And I feel like that's the way it needs to be done. Is like, just let the animal tell you what it needs.
Right.
And there we go. This idea about like ball pythons, everybody's favorite snake. This whole thing where they live in termite mounds, and that is why we can keep them in a rack tub. There have been many a study done on ball pythons. They've been done by the governments where ball pythons live, literally trying to figure out the most effective way to catch a ball python. Okay. It's a management perspective.
Right.
They absolutely come up out of the burrows. They're crepuscular. They hunt and forage. They move around. So, like, does that mean that it needs a 6 foot long, 3 foot tall, 3 foot deep enclosure? No. But does that justify keeping an adult female ball python in a little freedom breeder rack with no space whatsoever? No. So, you know, I just feel like we got to be a little bit more wise and educated, and also not be in these damn camps that keepers have a tendency to get into. And it goes for both sides, because a lot of people, you know, think that I'm 100% bioactive. I'm 100% naturalistic. I am not 100% bioactive, because bioactive can lead to problems if you don't do it right. But if the naturalistic condition means a rack tub with some aspen, then I'm keeping it in a rack tub with some aspen. So
going off of that, so I've recently had to downgrade my collection, so I don't have anything interesting now. But I do have a ball python, and I was keeping it in an enclosure. It couldn't fully stretch out in the enclosure, but it was large enough. But I wasn't sure if that was like, if that was good. But I had him on my porch, and then I was like, but the porch is completely screened in and everything, and I live down here in Southwest Florida, so I was like, well, let me just let him have the porch and see. And I put tarps on the side so that way he doesn't feel like he's completely exposed all the time and everything. And he was once I did that, so much more active. Like, he'd go all over the place, and he'd just find a little tiny spot he can shove himself in during the day and stuff. But at night and in the mornings, he'd be all over the place, so much more active once I was able to like.
That's a great point, because one thing that I think is really interesting is that a lot of people apparently don't go and look at their snakes after dark. Which I don't understand. I run around like a nut job in my house with my headlamp on all the time, and go creep on the snakes. I have them in my garage, and then I have them in my office. And so many snakes that are perceived to be not that active, and that's one of the reasons that justifies keeping them in a rack system or keeping them in a small enclosure. If you're up at like 2 or 3 hours after dark, they're doing laps around the cage. But because your behavior doesn't line up with their behavior, you make an assumption that they're inactive. My son I got 2 ball pythons for him because that's what he wanted. It's like, okay. And I don't sleep. So, you know, his are set up in vivariums. They're not in tubs or anything. But when I go in at 2 o'clock in the morning, they are always out. Like, I really honestly don't think there's been a time I've gone into that room when the lights were off where they haven't at least been, you know, slightly periscoped, moving around. And so I just always take it back to, like, I don't care about the camp you're in. Don't you just want to take care of these animals? Like, that should just be the main driver. It should just boil down to that. You know, it's that simple.
Yeah.
And if you don't have a cage that's big enough, or a room, let's be real, for like a 15-foot reticulated python, you probably shouldn't get a 15-foot reticulated python. I got the yellows because I knew they maxed out at about 8 or 9 feet. The enclosures they're in are 10 feet by 3 feet by 4 feet. And then I also let them come out and move around the garage. And the nice thing about my female, she's so damn big that I can just let her go wherever the hell, because she can't get anywhere. Like, she literally can't hide anywhere. So, but yeah, but to kind of keep this conversation going, the short tail python. So I'd read a lot about short-tailed pythons, and that's one where they said you got to keep them in a tub. You can't keep them in an enclosure. And so I tried keeping them in my PVCs, and there are absolute people that do, and good on you. My experience with them is there's something about a PVC. It probably has to deal with humidity in my house. That's why I don't think I can pull it off. I might be able to pull it off. I'm down where you are in Florida.
Yeah.
But they just failed to thrive. Like they stopped eating, horrible sheds. I moved them into a nice big rack system. I don't remember what the name of how big the tubs are. Maybe FB90s. And these are big tubs, and they're doing great in there for all I can tell. I let one of them out in my backyard just to see what it would do, kind of like what you did on the porch.
Yeah.
The thing slithered over to this part of my yard where the weeds were kind of growing up, coiled up, did this really cool behavior where it kind of worked its way down into the leaves, and the leaves actually kind of ended up on its back. And then I sat there and wrote a paper, like staring at it. It sat there for 6 hours and didn't move. Like
Wow.
it just, yeah, it was almost like, all right, this is what I'm supposed to do. So with, because of that observation, I have no problems keeping those guys in tubs. You see what I'm saying? So anyway, I just think that, and that's what I teach in my lab, is that if you're going to do this thing, that's the way you should do it. It should all be driven, learn about the biology, let the biology dictate what you do, and go on from there.
I forgot what I was going to say. It was, I forgot the question I was going to ask.
Yeah, it happens.
Yeah.
So let's go a bit back in the conversation, but when we're talking about, what true colubrids were, and you mentioned that it's like Asiatic rats and the Lampropeltis, Pituophis, and Pantherophis here in America, along with Spilotes. So what we say that's like an indication that the main origin or radiation point of true colubrids is maybe somewhere in like East Asia or North America then?
Yes. So natricids and colubrids immigrated to North. The idea, the theory is that they are they have an Asiatic origin. And then they basically came across this thing called Beringia, which is the great big land bridge up between Alaska and Russia. And I will fully admit, when I was actually working on the Dipsaddid book and I was trying to write up about the origins of Dipsaddids and all that, I kept thinking, like, how the hell do you get across an Arctic land bridge if you are a snake? Like, this doesn't make sense.
Right.
But that actually happened in the middle of the age we're in, the Cenozoic. And during that time period, the world was warmer and drier. So basically, that land bridge wasn't like a little bridge. It was a fairly expansive chunk of real estate. And it was kind of like equivalent today to like Ontario's weather. So super cold winter, but if you kind of made it across in the spring and made it down through the summer, you could get down into where we are. And one other colubrid I forgot, true colubridae, are the indigos. Drymarkin is in that group as well.
Oh, yeah.
Yeah, so, because I remember reading that I think a specific epoch is the Miocene. That's when the world started kind of cooling down, drying out a bit, got more open. I remember reading that's when we found a huge radiation of snake fossils. So I've actually heard people in the Miocene as the age of snakes. So I thought that was kind of interesting.
Yep.
So there's
a paper that just came out about that.
I had to look that up. So are there any Asiatic nutricines, like any remnant populations still left over in Asia?
Oh yeah. Oh no, there's not remnant. There are like full blown natricids, and they have the keelbacks. One genus is Rhabdophis, which includes like Rhabdophis tigrinus is the
poisonous,
pretty damn venomous thing, and it's the poisonous and venomous snake. Mm-hmm.
Is there a common name for that?
I know there's something over there called a tiger keelback. I don't know. I mean, tigrinus, that makes sense. Now I should get you hooked up with. There's a guy. He's wonderful. His name's Kevin Messenger. He came out of the Poly Lab and then he got his PhD. and he studied that system. So he's the guy that figured out, all of that. And now he's at a university in China, and his job is to basically like write books on the vipers of China and things like that. So he's kind of living the life right now.
I looked up. I asked about the common name because I looked up to see if there was a common name for that, just in Google, and like the thing that popped up was like Asian tiger snake. But if you look up Asian tiger snake, it's not the same thing. So I was like, that's weird. But I wasn't sure if there's a common name or not.
Nope, I would go with tiger keelback. And there's a bunch of other natricid genera over there, but I don't know too much about them.
Why do you know? Well, you just said you don't know like a whole ton, but do you know why the that snake is both poisonous and venomous? Like how? It's like how it evolved to be that way. Why haven't other snakes done that?
I don't know how the nuchal glands, those are the glands in the neck,
right,
how they actually evolved. Right. I understand a little bit about the venom poison system in that snake. Okay. They eat a lot of amphibians, and they basically are able to, in the digestive process, somehow take the constituent parts of the alkaloids in they're called tetrodotoxins and bufatoxins, right, out of the toads during digestion. And then they basically are able to send that up get it to the glandular tissue. And then the glandular tissue utilizes those biomolecules to then constitute a poison. And it's pretty crazy,
right,
bit of biology.
That's kind of like a snake version of a poison dart frog almost.
Yeah. Same thing with the formic acid with the ants and everything. Mm-hmm.
How come like hognose eats a lot of toads? How come they haven't developed a way to do that?
They were spending all their time just trying to deal with all the alkaloids. they have incredibly large adrenal glands on their kidneys, which are basically there to just deal with this tremendous load of toxins that they're getting. And a lot of snakes do some pretty cool things in response to feeding on toads. There's a species of Dipsatid in South America. It's actually it's a Phylodryas, which is the same genus that the Barron's racer's in. And these herpetologists in the early 80s were studying this whole group of Phylodryad snakes, and they were trying to figure out, like, how do you get 6 species from the same genus in one habitat? Like, I don't know, like, how does this work? And so they were really paying attention to what they were eating. And what they found was that this one species of toad, it's in the genus Rhinella, and the genus Rhinella is the same genus that the cane toad's in. So super huge parotid gland and really, really gnarly toxins. And what they found was that this snake would attack these toads, and the first thing it would do is it would flip them on their back and then eat them upside down. And the reason for eating it upside down, the biologists theorized, was snakes have a middle row, 2 rows of teeth that are along the palate of their mouth called vomeron teeth. And basically, if you're eating a toad and you're, working your jaws into it, you're going to be stabbing the damn parotid gland multiple times. And so if you flip it upside down, you don't have that vomeron tooth issue hitting the parotid glands. And then the mandibular teeth are oftentimes, they're more shaped like hooks, and they're oftentimes smaller than the maxillary teeth, which are the teeth up top. And so the idea there was those teeth are not only going to, like, they're going to be puncturing the glands less than the maxillary and vomeron teeth. So snakes have done all kinds of crazy behaviors to get around those toxins and amphibians. And in this case with the Asiatic guy, it was like, if you can't beat them, join them, and it just figured out a way to utilize the toxin for its own benefit.
Now, do you know are they like aware, if you want to use that word, that they have do they behave as if they have that poison?
No, they definitely do. They have stylized behavior where they kind of periscope and then they tuck their head down and they kind of spread out their neck, much like our water snakes do. Mm-hmm. I don't want to give the impression of a cobra, but it's definitely like, if you've ever caught a Nerodia, you know that if you get them by the tail and they're not chewing your arms off, they do that whole flare thing. and the rhabdophis will do the flare. And when they put their head down, the glands start to exude this white poison onto the nape of their neck. And the idea there is that if anything bites their neck, which is going to be where you're biting to kill the snake, you're going to get that toxin. And I think, if I'm understand it correctly, that it's very bitter. It does not taste good. It causes burning. Like, it's a true poison in that regard. And so the predators will spit the snake out before they finish it off.
That's really cool, actually.
Can you hear me?
Yeah, I can still hear you just fine.
Nate, did you have any?
Sorry, my mic disconnected. I didn't hear what you said.
Okay. Yeah, we can hear you. Yeah. That was really interesting. That was really cool. Mm-hmm. Did you have any other questions, Nate?
Yeah, I remember listening to your podcast. You mentioned that you chose, and by your own admission, made a mistake of doing your grad research with Eastern hognoses. You want to go a little detail about that, and maybe a little bit about Eastern hognoses in general?
So the genus Heterodon is both the bane of my existence and my favorite group of snakes. Like, I don't know how this has happened. In the field, they just absolutely kick my ass. I can find the rarest animals on earth, but if I go on a trip and I say I'm looking for Heterodon, now, in the back of my brain, I put it at about a 1 in a million shot of actually finding them. This all started with my master's. So with my master's thesis, I got in the poly lab. I told Dr. Polly I wanted to study a snake. Dr. Polly immediately retorted, as a good advisor should, snakes are one of the most difficult groups of animals to study because you can't find enough of them to do anything with them. What I should have done in that moment, and I didn't, and now I'm doing it as a professor here, is I should have said, well, I'm going to study queen snakes because I've got this thriving population of them back home. But for some reason, I didn't say that. And I said, well, I would like to study a snake that nobody's looked at in West Virginia. And then Polly said, well, here they are. And he listed off a bunch of snakes. And of course, one of them was a hognose snake. At that point in time, I had not herped a hognose snake, not found one. But he also said that he knew where there were a bunch of them around this town called Beckley in West Virginia. And Beckley was like an hour and 15 minutes from Huntington. So I was like, I'm on it. So I wrote a grant, got funded through the DNR, and my thesis was supposed to be, 1, determining the distribution of hognose snakes in West Virginia. And I was actually in the end able to do that. It took me well beyond my master's to do it, but I'm stubborn as hell and didn't give up until I had enough data to say that. But then 2, I was going to do an audicology study where you basically study a little bit about a lot of these hognose snakes on this abandoned mine. And I will keep you from the nitty-gritty details, but this man named professor, he's actually a herpetological god. He's my hero in herpetology. Henry Fitch, he was based out of University of Kansas. He studied snakes by setting up drift fence arrays and putting down traps. And they're called box traps. All it is if you take a minnow trap and you turn it into a box, it's a box trap. It's an inverted cone trap. And so I thought, I'm going to do this, the way Fitch did it. So I begged, pleaded, stole, did everything I could to get grad students to help me set up over a mile of drift fences on an abandoned strip mine in southern West Virginia. And I built over 100 of those traps, and we put them all up, and no hognose snakes to be seen. And when I got to the mine, I just kept thinking, like, this doesn't look like hog habitat, but this guy had told Polly they were there. And we were 2.5 months into the study. No hogs. I'm starting to get stressed out. Found all kinds of other snakes. And I met the guy on the mine. And there was another grad student with me. Her name was Jamie. And I had my stump ripper in my hand. And if you don't know what that is, that's a titanium tipped snake hook. It's important for the story. And I walked up to this guy who We're not going to say his name. And I said, hey, are you the guy that said that the hognose snakes were here to Dr. Polly like about a year ago? And he's like, oh yeah, that was me. And I said, well, where did you see them? And I'm basically trying to get more information out of the guy. And this guy's kind of older. And he looks at me and he goes, yeah, but I haven't seen a hog here in like 20 years.
Oh my God.
Like, 20 years? What do you mean 20 years? And he goes, oh no, I saw those like back in the late 70s, early 80s. I guess I forgot to tell Tom that. Polly's name was Tom. And I had the stump ripper in my hand, and I'd put in hundreds of hours at this point in time. There was a 5-day stretch I did not leave that mine. Like I was just on the mine. And I apparently like lifted the hook up to take off his head with it. And Jamie just like gently put her hand on my hand and pushed the snake hook back down. But in that moment, I realized like, I'm not going to get a degree. Like, I was right. There are not hognose snakes here. What the blank is going on? So I was able to pivot. Thank God the DNR was understanding. And my thesis, like within 24 hours, it had shifted. And I wasn't studying eastern hognose snakes anymore. I was studying reptile and amphibian communities on an abandoned strip mine. And it's kind of hot garbage because I went into it, with the objective of studying but luckily I did what my advisor told me, and I took data on every single reptile I caught. And I'm so happy I did, because I had enough to pivot to that thesis, but I didn't really have anything to do with statistics. So it was very descriptive. And it was still interesting, but that's what like started this interesting dynamic I have with hognose snakes in the field. Last May, I went up to Pennsylvania. There's hognose snakes are relatively common in the Poconos. And for the book, I was like, all right, I'm going to go find them. And this is the best place to find them. And I had a spot where the people that gave me the site were like, we do not go and not find them. And I don't know if you remember, Nate, this wouldn't be the case with you, Matt, but in May last year or this year, we had this weird weather where it was supposed to be like a really nice, cool weekend. And then we got this sudden upwelling of weather from the south. And we had a weekend where it was like 100 degrees in May. Yeah. That was when I went to look for hognose snakes. So it was a burning hellscape on this railroad track. So I didn't find them there. Now I did find them though. I pivoted and went back to my blessed West by God. And I have a friend in the Eastern Panhandle, and I called him and was like, please, for the love of all that's good and holy, I have to have a win. Can you just like help me get them? And it ended up being the best West Virginia herp day ever. And we did get them. We also got corn snakes in West Virginia, and that's like the rarest snake in West Virginia. So, but then we did this giant trip out west, 10 days, literally went in October, Hogtober. Everybody talks about Hogtober. Found over 100 snakes. It was a great herping trip.
Wow.
We did get 3 Easterns. They were all dead on the road, but I do get to say I went to Kansas and saw Heterodon. They were smashed to oblivion, but I did see Heterodon. I just didn't see Nassicus. So I have plans to go out to the Midwest again. This time I'm going to shoot for May. And then and the other thing that's really funny is Simis, the southern hognose snake. I had to make the decision, am I going to go for Simis this year, or am I going to go for Nassicus? And I went for Nassicus. And while I'm out in Kansas looking for Nassicus, Kevin Messenger, the guy I talked about, who's in China, he works on southern hognose snakes. And he messaged me while I was out there. It's like, hey, can you get down here? I was like, why? He was back home in North Carolina. He's like, well, my friend just sent me these. And he sends me pictures of 4 freaking southern hognose snakes the guy found on the road I would have been in had I made the decision to go down to look for southerns this year instead of western. So like it literally is this like almost running joke with myself that like it's just like in the ether that I'm not going to find them. So
I had a high school teacher who's just a naturalist, local really into like local naturalist stuff like that. Spent all of his childhood and adult life out in the woods. And he said in all his 60+ years of life, he's only seen one wild eastern hognose snake, only one time.
Mhm.
And the only other time he's ever seen a hognose steak was one of his students. He and his dad found a hognose, and of course, because we're in Ohio, everyone has copperhead phobia like
Yeah.
crazy. So they instantly killed it, put in a jar of alcohol, labeled it the deadliest snake in Ohio, and said, "Hey, look at this copperhead we found." And he had
Yeah,
to
that
very
happens.
gently put them down about that.
Yeah.
Me
So
and
I'd double check. But I'm pretty sure eastern hognoses are actually state endangered here. But I'd double
They
check
probably are. They're a weird snake. They are what we call locally abundant. So if you get on them, they're kind of common. And as you go into the southeast, there's definitely areas where you can find them. I'm really it's kind of funny. They're running one of the other jokes of the lab is that we got this South Carolina grant, and I am just as happy to go to South Carolina to look for the herps as I am the crayfish. So it's kind of a win-win. And where we're doing that work, there's a lot of INAT records of eastern hogs. But if you're not in one of those areas, they're just a pain in the ass. So
me and Nate, our senior year of college, along with another buddy of ours, came down here to South Florida um, to go herping and fishing and whatnot. But also, we were hoping to catch a Burmese python, and so we were walking around the Everglades and everything. And I don't know if I ever told you this, Nate, but we like a few days after we left there, and the same exact spot where we were standing, a 14-foot long python was found in the same exact spot we were at.
yeah, that's how it always goes. No, but this is 2022 is gonna be my year. I'm not giving up until I get the Nassicus is personal. Like that Kansas trip was without question in my adult life the most dedicated trip I've done for herps, and might have even been the longest herp trip I've done. Period. As far as consecutive days, and just like you said, we got home and I had a friend, Curtis Schmidt. He was the collections manager at the Sternberg Natural History Museum in Hayes, Kansas. And we actually were landing. I was walking out to the car and Curtis sent us a message. He was like, guess what they found where you were driving? And it was a whole bunch of Western hognose snakes. So it's just written in the stars, man.
Yeah, this isn't reptile related, but it's the same exact thing with a garfish. I do a lot of fishing, and a gar is just this white whale. And every single time I get closer to catching it, and every single time I think I can't possibly get any closer to catching it, the next time I have to actually catch it. And I mean, just last week, I had the fish all the way up to the shore. I went to pull it on, but the line broke because of the way the gar are at. But there's like a bed of seagrass, so it was still sitting there, or just water grass. And I dove in the water, and I had the fish in my hand, and it's still because it's a fish, it just squirted out. But it was like a 2-foot gar, and it squirted out. But I had it in my hand. I was so mad. I was like, "You got to be kidding me!"
Yeah, no, been there many, many times.
Yeah, that's how it is, though. But it's fun. I still enjoy it.
Yep.
I have friends that are when they go herping or fishing, they have to catch something. I just enjoy being out there. I mean, I want to catch something, but I just enjoy being out there. Like I'm perfectly fine with not catching anything. It's just I just like being outside. So
no, totally understand that. I'm more that way now that I'm older than I was when I was younger. When I was in my 20s and 30s, it would feel like a failure. Like this trip to Kansas, it might seem like I feel like it was a failure. It was not a failure. I got all kinds of habitat pictures, and then I added so many animals to my life list, it was ridiculous. So like it was nothing but a win. It's just funny that I went all that way for one critter and literally caught everything but it.
Right,
So,
exactly.
yeah.
Now I've been there just going for one specific thing, and you have a great day, so you can't complain. But
yeah,
yep. But you didn't catch the thing that you want. Yeah.
Yep.
I've been trying to catch red-headed agamas down here in South Florida, specifically the males, because the males they just look cooler. But it seems that whenever I find a population, there'll be like 2 males, and there's usually like a brightly colored one and a less brightly colored one, which I'm assuming is the dominant and subordinate male, and then a large swath of females. And the females are much easier to catch, probably because They don't seem to be as fast, but it's also probably just because there's a lot more of them. So
Yep.
So I keep catching the females, and I've been trying to catch a male, but I haven't been able to so far. That's been kind of my white whale with reptiles so far.
Have you tried to noose them?
Yeah. It so there was there's a population in front of this Walmart, so I kind of look like an idiot trying to like grab this thing in front of this Walmart.
Mhm.
But it was in this bush, and it kept like running around in this bush. So I'm trying to noose it, but I used two different nooses. I use a paracord, and then I have like fishing line, just depending on what I'm trying to catch. And I had the paracord on at the time because I was catching an iguana when I first saw it. And so I was trying to get with the paracord, but I couldn't get anywhere near it, and it'd run. So then I tried with the fishing line, but it was just too windy, and I couldn't. And because of the way the bush was and it was so windy, I couldn't get it. And then the only other, so that was over in Miami. When I first moved down here, because I moved down here August of last year.
Gotcha.
When I first moved down, I couldn't find. At first, the only populations I could find were over there in like Miami area. I'm starting to notice more and more, especially now that they're actually closer and closer to where I live. So then I started finding them in Naples. Now I'm finding them in Fort Myers and stuff, which is a lot. Not anywhere where I thought I'd find them, but there's this gas station in Naples where they're at. But I can't. The area is so open; they run away too quickly. I can't noose them. So I just have to try and get him with my hands and stuff. Which I came really close one day because he's in a bush and he thought he was hiding, but I couldn't exactly see where he was at, and I reached and I just missed him. But
yeah, I haven't had much experience catching lizards. I took a bunch of kids down to Key Largo on a diving trip, and there were iguanas all over the place. And when I lived in Florida, I lived up in Melbourne, which the Iggies weren't up there, but they were definitely, they could be up there now, but they weren't very common, at least. But they were everywhere down in Key Largo, and I went up into a mangrove tree and caught one, and it was really funny because I was expecting, when I put hands on it, for it to just be a total explosion of animal, and it just totally gave up. Like the second it just went limp. In fact, I actually thought, like, did I break its spine? Like, what the hell is going on here? But yeah, have you gone down into northern Key Largo where the tokay geckos are?
So the iguanas, first off, the iguanas in Key Largo are huge. Like they're way bigger than the ones that you'll find in Miami or Naples or anywhere else. They're huge. And that is funny. When you did you noose it or did you grab it?
I was dangling in the mangrove tree and just grabbed it with my right hand and hoped for the best. So
it's really funny. Whenever I noose iguanas, when I grab it, they twist and turn and go crazy. But as soon as you put your hands on it, they are completely like, they don't move at all. It's crazy. So that's interesting that you say the same thing. But as soon as I grab them with my hands, they're limp. They don't move at all. So it's pretty interesting. I'm not sure why they do that. But yeah, when we took the trip, me and Nate and the other guy down, we stopped in Marathon, which is about halfway through the Keys, and under a bridge. We were going to sleep there. We kind of hoboed in a van for like a week, and we were going to do some fishing that night. So we're setting up our fishing stuff, and we hear this weird noise. And we're all like, what is that noise? And then me and Nate, because our other friend's more of a fishing guy, he's not as much of like a herp guy. Me and Nate just look at each other, our eyes like widen, and we're like, it's a tokay gecko. And so we go running after it. But so there's like a line of trees there, and we can hear it somewhere in the trees. We're looking for it with our flashlights. But there's a business on the other side there with a bunch of boats and whatnot. And the guy's just like, what the hell you guys think you're doing? We're like, looking for a lizard, a tokay gecko. And he's like, is that the best excuse you can come up with?
Yep. Sounds like you're herping.
Right, exactly. So we were just like, apparently he's been having kids breaking into his stuff, so that's what he thought we were doing. But I'm sure you've been in that situation where you have to explain to someone that you're herping, but they have no idea what you're talking about, or why would you even be doing that? So it was pretty funny. We ended up not finding it. The guy was kind of like, crappy about it, so we ended up walking away. So
They were all over the neighborhood where our house was. And so we were doing what you were doing, just with flashlights at midnight around people's houses. So, but nobody came out and bothered us. But we ended up getting a bunch of them. They were all
Really?
over the place where we were.
In
I
the
heard
north
them
part of
first, like
Key
you
Largo?
did. What was that?
In the north part of Key Largo? Key Largo?
Mm-hmm.
Interesting. I have to take a trip over there and check it out.
Yeah.
That'd be really cool to find a wild tokay gecko.
Absolutely.
The Mediterranean ones are everywhere here, but
Mm-hmm.
that'd be cool to find the tokays.
Yeah.
The day geckos are supposed to be down in like the Keys too somewhere. I've heard, but I've never seen one. So
No, I didn't get any Phelsuma. We got those, and then being the Nerodia guy I am, we got a salt marsh snake, which was pretty cool.
yeah, that is cool.
Yeah.
Yeah. The overwhelmingly the vast majority of snakes I find here are like banded water snakes and black racers. There's like
Mm-hmm.
if I see a snake, 99.99% of the time it's one of those two. I just found one last night actually. I was actually frogging, but I found some snake or two, so that was fun.
Nice.
Yeah, I was just at that Columbus Expo last Saturday. Of course, I saw plenty of tokay geckos for sale for not that much. Unsurprising.
Mm-hmm.
Definitely imports. But also, I saw a ton of fire skinks as well. So I'm kind of interested in. Now, also my interest is spiked in maybe starting a fire skink colony of some sort. But we'll see.
Yeah, fire skinks are cool. I had those when I was in college.
Did you like them? Because from my understanding, they don't. You don't see them a lot. Like they're always burrowed and stuff.
They burrow if you have a light, so they like to bask, and so I set my lights on timers, so they would basking light would be on an hour and a half, and then it would be off, and then a basking light would pop up somewhere else for an hour and a half, and by having the lights, it almost forced the animals
to move around,
to get out, and that way, I could actually see them doing stuff.
That's really smart. I have to look into doing something like that.
What type of enclosure did you keep them in?
They were in a 40-gallon breeder tank, so nothing overly crazy, but no, that's what I had them in. And then I had a bunch of cypress mulch and then some rocks and stuff like that for them to burrow under.
Yeah.
I had them for about a year and a half. I think I gave them away when I went to Marshall, actually.
Mm-hmm.
Yeah, I saw them for sale, and they weren't that expensive either. I think they were like, could be misremembering, but I think they're like $15 a piece, which...
Oh, man. I saw those same skinks because I was at that same expo.
Yeah. Yeah, I was talking to Andy when I was there, and he said, "Oh, you just missed him."
Yeah, I had to get... I am having a hell of a time with my garter snakes. I have the Oregon red-spotted garter snakes, and I have these 4 females that are adults and perfect. And the zircles are kind of giving me and helping me with the garters, and they've given me some males, and the males just keep, for absolutely stupid reasons, dying. So I had to... we're right at brumation time, and I asked the zircles if they had any surplus males, and they said they did, so we were going to do a trade, and I had to get those, but then I had to also go shopping with Kathy for Christmas, so it was like a rocket run from Wheeling to Columbus, which is about 2 hours. I think I was in the expo for like a half an hour. Then I just turned around and went all the way back to Wheeling so I could get home by noon. But anyway.
Yeah.
All right. Well, Nate, did you have any more questions?
Not that I could think of off the top of my head. Unless you have something else you want to say.
Yeah. Did you have anything else you wanted to mention?
No. I love what you two are doing, and I really like that there's podcasts now for all the reptile nerds. Yeah, and I love the concept of yours where you have the science, the field stuff, and the husbandry all intertwined. Because in all actuality, that's more reflective of me than my own podcast. So, so no, keep doing the good work, guys. It's a good one.
Oh, thank
awesome.
you. I'm glad.
Thank you. I really appreciate it.
Yep.
All right. Well, until next time, signing off.
Thank you.
Bye.